Good morning, everybody. I'm Lionel Imbert from Instruct Center in France, in IBS in Grenoble. Welcome to this Instruct-ERIC Structure Meets Functions webinar, the number 37. We will have 3 presentations today. The first one will be given by Catherine Megerl from, she is coordinating the work package on staff exchange between EU and LAC in the EU-LAC ResInfra project. Please, Catherine, let's go.
Thank you, Lionel. Thank you very much. So hello to everybody. Good afternoon to the people listening here from Europe, and good morning for people or participants joining from Latin America. So I would like to briefly introduce the EU-LAC ResInfra Plus project, which I'm coordinating, and Instruct-ERIC is a partner in this project. So therefore, thank you, John, for the invitation and giving me the possibility to just briefly present this project. So ResInfra Plus is a Horizon Europe coordination and support action. We have this project running for about 2 years. It will end in December this year. We have a budget around about 750,000 euro. And the interesting part is we have a consortium of 18 partners from different countries in Latin America and Europe. And this is a follow-up project from the last ResInfra project phase, which ran from end of 2019 until April 2023. So there is already a lot of experience and cooperation going on between the 2 regions. And so therefore the project is coordination and support action. It has a main goal which deals mainly with establishing a sustainable framework for bi-regional cooperation in between research infrastructures in Europe and Latin America and the Caribbean. And so we have a couple of different steps and activities we're going through to meet this goal. And okay, there's as a starting point, of course, the wish to increase visibility of research infrastructures, especially in Latin America and the Caribbean, and also share good practices in research infrastructure cooperation. And this we're doing through also a series of webinars and online talks and policy dialogues between the 2 regions. But we organize also staff exchanges and conferences, and this would be like the concrete level of infrastructures themselves. And then the other level we're talking to is the political level, and here especially the EU-CELAC Joint Initiative on Research and Innovation, the GIRI-SOM. There's a senior officials meeting every 2 years, and ResInfra Plus informs and is recognized by this policy dialogue. And then there is also a bi-regional working group on research infrastructures, and they're also working on this political level. And the idea behind this is of course, proof that the usefulness of the bi-regional RI cooperation. So here's a brief overview of the mentioned 19 institutions. We have 6 European countries and 8 Latin American countries participating. And the interesting part is also that we are a mixture in the consortium of funding institutions and but also ministries. And then we have these 4 European research infrastructures, which are partners in the project. And we have here on one side, of course, Instruct-ERIC. We have MIRI. It's also NERIC. We have RICAP and CIEMAT from Spain. And we have LifeWatch-ERIC, which is also based in Spain. You see there's quite a range of areas covered, thematic areas covered by these infrastructures. But as I said before, it's a good mixture between having the political level and also the concrete world of research infrastructures cooperating directly with each other. So we have this different activities foreseen for this last year of the project. We are working on a dashboard for representing infrastructures in Europe and Latin America. We are organizing, as mentioned, these workshops and the policy dialogues. We have 5 thematic priorities. We have health, food security, biodiversity, climate change, energy, and information communication technologies. We are working on a cooperation report and having like a focus on this mission-oriented innovation activities and the role of research infrastructures within this innovation format. And then work package 4, which I'm working in or leading, is these different webinars and staff exchange programs. And finally, we have planned to have a huge online stakeholder forum in October this year. And then, of course, we have dissemination and communication activities. So just to give you a small overview on the results, not only from the current project, but also from the first ResInfra project. We are working on this dashboard, and it's a constant update on institutions in both regions, in Europe and Latin America. And we have then this analysis and cooperation report. We have these activities where we really strengthen the cooperation on the level of the institutions. And then the goal and the idea is also to contribute to the update of the strategic roadmap between Europe and the CELAC region. There is a pillar on research infrastructure, which is again the political level. And there is an update plan for the years 26 until 28. And then we are also present in a series of platforms and initiatives. As I mentioned before, the joint initiative on research and innovation between Europe and CELAC. There are also conferences like ICRI, Carla, and we're also providing information on funding. There's a joint call from the EU-LAC research interest group. Some of the partners have been successful applying for funding for this bi-regional cooperation. And then, of course, we work together with other European Horizon Europe funded projects, like in the past RIVIS, Sincere, EU-LAC Permit, etc. So that's it. Thank you very much for your attention. And of course, there's the website. I think John already posted it in the chat, and we're on LinkedIn and X also. So thank you.
Thank you, Catherine, for this nice overview. And if you have questions for Catherine, please use the Q&A sections. And we will follow with the presentation of Eugenia Flores Giubi. And sorry, Catherine will reply to you by the Q&A section also. We follow with the presentation of Eugenia. Eugenia is a Paraguayan biochemist specialized in secondary metabolism and biochemistry of plants and microorganisms. She obtained his PhD in Spain, and she works now in Uruguay. If you have, we have a presentation of 20 minutes from Eugenia, and if you have questions, please use the Q&A sections, and I will read your question for Eugenia after that. Please, Eugenia, it's up to you. We don't hear you. You are mute.
Here I am. Hello. Now, yes. Okay. Can you see my screen now?
Yes, perfect.
Okay. Hello, everyone. I'm Eugenia from Paraguay. And today, please, you can, first of all, I want to thank for the invitation to participate to this webinar. I will speak a little about the role of secondary metabolites and proteins in a specific fungi that called Macrophomina phaseolina. And first I will make a brief introduction, and then I will show you our results, and maybe we can find something interesting for a proposal in the next years, maybe soon. I hope. Okay, we recognize this from the One Health concept, where we know that the interrelationship between healthy food, humans, and soil, and interlinked with a microbial loop is very important for the maintenance of the human public health. So everything or anything that could impact on this, in one of these parts, we can have some troubles to solve here. Today, I'm going to speak about a fungi called Macrophomina phaseolina. It produced the disease named charcoal rot. We're working with the phytopathogen that was first mentioned in 1970s. This phytopathogen caused a disease that is spread worldwide. This fungi has a lot of hosts, almost 100, maybe more. And for example, corn, cotton, sesame, sorghum, sunflower. And it is known as a global crop destroyer. For example, here we have the geographical distribution of this disease on mice. And you can see also that we have symptoms that we can see on every part of the plant, a plant like flower, leaves, stem, and roots too. In the United States, is the second cause of important economic losses, and is well described for this, and the research is increasing in this area for this reason. And if we can see a little more inside this phytopathogen and the symptoms, when we can see that the wilting symptoms on young plants, and the symptoms in roots or stem, and the symptoms in seeds too. This example is in soybean seeds. That is an important source of food for worldwide. If we see a little more, a closer look in vitro dishes, we can see the growth of these microorganisms, like gray colonies. So we can see two microsclerotia and conidia that are produced in a specific type of culturing media. We know we have a lot of information from this phytopathogen, but we have questions too. We don't know yet the exact mechanism of action, and obviously, if this microorganism is so successful in the environment, it has to produce some molecules that make it able to do that. So, for the first place, I can show you the presumed events at molecular level. We know that the microorganism is in soil, so it penetrates to the plants from the roots, and it has a lot of enzymes and effectors and phytotoxic metabolites, and that all molecules can alter the hormonal system. And in the first place, it has a biotrophic phase, and then switch to a necrotrophic phase. We don't know exactly what are the signals to change this stage. In Paraguay, this is my country, we could took samples in this region in Paraguay. We have now like 10 or 15 isolates from this phytopathogen. And the question we have now is, what are the phytotoxic metabolites? Which proteins are involved in the pathogenic process? And of course, if that molecules can have another activity that could be important for human health approach, like antifungal or antibacterial activity. This phytopathogen is known because it has a large genome, but only few genes related to secondary metabolites. And we suppose and we know that there are several metabolites that we don't know yet. And that kind of metabolites that are silenced, we know as cryptic metabolites. And our objective is to express them. And one approach that we are taking is the OSMAC approach, called one by OSMAC is one strain, many compounds. In this approach, we modulate the in vitro conditions to obtain cryptic metabolites. We select one of the strains, the more phytopathogenic one is FCQ11, because my institution is Facultad de Ciencias Químicas, so we are very creative. The culture of the fungi was carried out in two culture medium, PDV and Sapecdox, and we employed soybean leaf infusion to enrich the media, the culture medium. And then we also check the phytotoxic activity of these molecules, the secreted molecules, in two bioscience models. One is the necrosis induction in soybean leaves, in disc of soybean leaves, or in seedlings of soybean. And we saw that in the culture medium with infusion, PDV plus infusion, we saw a higher level of necrosis of the molecules that are secreted on the culture media. And we also saw different phenotypic and morphological changes on seedlings compared with control. We isolate and we detected and isolate a few metabolites. One of them is melanin. We characterize this metabolite by NMR techniques and another spectroscopic techniques like mass spectrometry, and we are supposed that we are very sure that we have melanin in our culture media secreted by this phytopathogen. Also, metabolite biosynthetically related to melanin is 4-hydroxymelanin, also by NMR techniques, mass spectrometry detected and confirmed. And we don't know exactly the mechanisms of action of these molecules. It's not the first time that we found this in this phytopathogen. We have reports earlier that report first these metabolites in Macrophomina fasciolida. And then if we can see the chromatographic profile of this is the secreted molecules on PDV, and here we have in PDV plus infusion, we saw different profile, chromatographic profile. Without infusion on the culture media, without the stimuli for the production of metabolites, for example, we have melanin here, and it switched the ability to produce molecules. So we have more types, we have more diversity here, and it appeared botrodiploin, it is a metabolite that is known for its phytotoxic activity, and hydroxymelanin 2. Here we have other metabolites besides these three that I mentioned. We have also aspergillin, isoaspergillin, and acetylphomalactone, and these are only a few of all the molecules that these fungi can produce. In the literature, we know these fungi could produce other ones like tyrosol, succinic acid. All of these related to the phytopathogenic process. We are trying to isolate and detect more of these compounds, but we have these 6 already. We tried this on the bio side with foliar tissue, and we saw the phytotoxic activity by inducing necrosis of all of them. You can see that all of these molecules can produce these fungi, and others that we don't know yet, at once. So this is a very, very, very aggressive phytopathogen. So we are trying to produce this on a major scale to isolate more of these compounds, to carry on experiments of gene expression and interaction with several types of host. For the other hand, we continue the work and make a proteomic approach. We characterize in silico and by experimental data about the secretome of this phytopathogen. So we could detect a total of 250 proteins that are in the secretome. All are secreted by this phytopathogen, and most of them are enzymes like caseins or peptidase, and others that would be effectors, for example. So the predicted effector proteins were also found that would induce plant cell death or suppress plant immune response. One of the proteins that we are trying to focus on is the necrosis and ethylene inducing protein, NEP protein. This is a known protein that we detected. We not detected on culture in PDV in vitro, but when we did some infection in root, we detected 4 days post inoculation, and we already carry out an experiment of heterologous expression of these proteins in Nicotiana bentamiana. The NEP1 is a family of proteins, NEP1, 2, and 3, and you can see here the effect of the overexpression of the protein. We are not focused on isolate the protein and working with these pathosystems to know the genes related to the pathogenesis and characterize them. The NEP protein as I said before, is a known protein related to the interaction between pathogen and all the cascade of events that conduce to local necrosis, spreading lesion, and of course the response of the host that has to respond to the phytopathogen and control it for survival. And for that, I'm finished. The second question is related to the bioactive molecules that are secreted from this phytopathogen. And we have some interesting results about the interaction in between these secreted metabolites against Candida parapsilosis and Candida albicans. You can see here is like the same response as the control, that is fluconazole. You can see here a halo of inhibition with Candida parapsilosis and Candida albicans. We are not now trying to identify these bioactive molecules and, of course, their mechanism of action. And we think that it will be very interesting to collaborate with some groups of that you have over there. This is my chemical biology group. She's my boss, Cristina Romero. She's listening now. And my team with Dr. Barua, and we have 3 PhD students. They will be glad to join your groups and work with you and in some kind of collaboration. And Daniel, Jasmine, Alberto, and 2 master's degree students, Mamiliana and Linda. And just for finish, thank you very much for your attention. And before I conclude, I would like to sincerely thanks to the invitation to Grenoble, France. First, I had the opportunity to know the impressive infrastructure you have there. And I have the privilege to know very, very interesting and wonderful and kindly people. Thank you very much for the opportunity. I'm open for questions. Thank you.
Thank you, Aurélien, for your presentation. I look on the Q&A section for the moment. I have no question. Don't hesitate to ask your question in this section, please. For Aurélien. Maybe I can start. And it was nice work. And what are your wish, if I can say like that, to continue your work? Which in a technical aspect? What do you need from a European or Latin America platform or facilities? What can you expect from us?
Okay, first the spectroscopic infrastructure first, and then we need help to characterize the proteins and the target of these proteins in the pathosystems. And with Candida albicans, we suppose that we will have a pool of metabolites or maybe peptides or proteins that could inhibit the growth of these human pathogens. So we really appreciate to collaborate in this area, in Candida albicans especially.
Okay. Can you see also the Q&A section, Aurélien? If you,
yes, I can see, but
I have nothing on my side, I think. If you want, you can
see anymore.
Yeah. If you have question, you can continue to ask in this Q&A section, and Eugenia will reply to you in this section also, and we can continue with the next speaker. I will introduce Professor Gonzalez Olvera. He obtained a PhD degree in advanced technology with a specialty in molecular biophysics by the National Polytechnic Institute in Mexico. At present, Professor Gonzalez performs basic and applied science research and teach science and mathematics at the Tecnológica University in Querétaro in Mexico. His research interests are mainly focused on biophysics of nucleic acids and composite biomaterials. Professor Gonzalez have a collaboration with the CEITEC in Brno in Europe since 10 years now, and particularly with the Dr. Radovan Fiala and Radek Marek. Please, Julio, we listen to you.
Okay, thank you, Lionel, for this introduction. Hello, good afternoon, everyone. I'm Julio César González from the Tecnológica University of Querétaro in Mexico. Then let me share with you my presentation. I'm going to share here. I hope you
Perfect.
see. Okay, perfect.
Perfect, yes.
Okay, then I'm going to start with the presentation. Again, my name is Julio César González Olvera from the Tecnológica University of Querétaro, Mexico. And I'm going to present today the project titled Studies on the Protonation of 5-methylcytosine groups in double-stranded DNA polynucleotides. First, I would like to thank to the Instruct-ERIC program for inviting me to present at this webinar. So then I'm showing you the agenda for today for this talk. I'm going to start by talking about the biological relevance of both the post-traductional and post-transcriptional modifications of cytosine groups in both DNA and RNA molecules, respectively. I mean the formation of 5-methylcytosine in these nucleic acids followed by some significant findings reported in 2 different papers related to NMR, nuclear magnetic resonance, and UV absorption, monitoring of methylation in cytosine residues. Published in collaboration with Professor Radovan Fiala and Professor Radek Marek from CEITEC in Brno, Czech Republic. Then I will share you some experiments we have planned for a new related project, which is the continuation of these previous studies. Finally, I will show you a few preliminary results obtained by UV absorption titrations on new DNA sequences, along with the presentations of the conclusions, final conclusions. Okay, so I will start with the introduction of the biological relevance of methylation at 5 sites of cytosines in DNA and RNA. This is a 5-methyl cytosine molecule, which is a critical modification on DNA and RNA. So some reports in literature are the following. This 5-methyl cytosine is found in controlled regions in DNA by Watcher et al. At 2014. And we can see here in this black dots, different positions of this 5-methyl cytosine in 3 different cell lines of mouse stem cells. Also, this 5-methyl cytosine has been found in some regions. We have contact with or are related to repression of gene expression. For the in DNA for the expression of genes. So these DNA modification have been found in gene bodies, as was reported by Erich and Lacey in 2013. And these modifications inhibit the binding of the transcription factor CTCF, or 11-zinc finger protein, which is involved in the regulation of the 3D structure of chromatin, affecting the cell differentiation processes. Regarding to the RNA, 5-methyl cytosines are abundant in different RNA molecules, such as transferase RNA, ribosomal RNA, and messenger RNA, affecting their stability, assembly, and translation respectively. Additionally, RNA methyltransferases mutated with 5-methyl cytosines are associated with diverse pathological processes, such as nervous system disorders and cancers, some kind of cancers. Finally, in this introduction, the methylation at the 5th position of cytosines is relevant for long non-coding RNA because it promotes tumor progression, as we can see here in this slide. And also, this is done by controlling the expression of critical oncogenic drivers in a specific cell carcinoma. However, the study of this type of methylation in long non-coding RNA is still in its initial stage. So this is a brief introduction about the biological relevance of this methylation in cytosine base roots. So now I will present you some previously reported results, which are the basis for our proposed new project. Initially in 2018, also in collaboration with Professors Radek Marek and Rad Vanciala and my partner as a student, Matusz Turec, we observed the end part of a transition here, indicated by the rows, attributed to the protonation of central 5-methyl cytosines, these ones, for the black squares, and this one for the red circles. In the corresponding base pair, this was done in DNA duplex molecules, with 17 base pairs, and monitoring by UV absorption titrations at low temperatures and low condition salt conditions. This initial finding suggests the possibility of protonation of those residues, 5-methyl cytosines, around neutral and physiological pH value. This is important for the biological relevance. So the protonation of other cytosine and adenine residues in these double-stranded DNA models occurs after pH 5 as was expected. And then the double-stranded molecule is dehybridized or separated to the single-stranded situation. This was reported in a paper in 2019 in collaboration again with our collaborators in Brno in Czech Republic. So also as part of this work, we do some NMR experiments, nuclear magnetic resonance experiments, and I'm showing you here the immunoprotons spectra of the 1 in 17. This one is that is paired with the 5-methyl cytosine in a central position of this double-stranded model. And interestingly, this base pair don't disappear until pH 3.9 over here. I mean, during all this acidic titration, the base pair is still formed. It does disappear, indicating that that the 5-methyl cytosine is protonated, as we can see here in this position, and the base pair is remains. This suggests that a base pair with positive charge is highly probable to be present in double-stranded DNA, maybe in physiological condition, with most of the positive charge in the O2 position over here, located in the major groove of the double helix, affecting, as we can try to prove, the interactions of this part of the DNA with other proteins, for example. So the next finding we consider is interesting is the effects of protonations on protons in other residues. There's a cross relation ship effect of the protonated 5-methylcytosine with other residues, for example, the thymine 5, which is bisonal to this protonated residue in double-stranded DNA. As we can see here in this aromatic protons spectra, we conducted in Bruno in the first visit we have to the CEITEC. So this is an interesting result because we are observing that some of other residues in the double-stranded models are affected by these protonation processes. And this is a result we want to study deeply in our proposed project for this year, and I don't know, maybe the next year. So this is the background, and this leads us to the objective of the proposal, which is to determine the physicochemical and structural state of the protonated cytosine and 5-methylcytosine bases in the corresponding base pairs included in model polynucleotides and biological DNA. We are going to extend these studies to longer molecules of DNA and even RNA, examining the effects of physical conditions, as we saw later, by experimental techniques and computational algorithms. So we have performed some initial size here in Querétaro, and we have planned to continue some NMR experiments the next summer. Not by me, but by collaborator, Professor Reinaldo Ples, will be, I hope, will be in Bruno in CEITEC with Professor Rado Banfiala for the next summer. And the initial assays we have done here in Querétaro in the last few months are related to the examination of size, ionic strength, temperature, etc., on the protonation of this residue of interest, and also the native cytosine groups in single-stranded and double-stranded oligonucleotides of 13, 17, and 21 nucleotides. We are now tried in longer models, even 17 or 21 nucleotides or base pairs for the double-stranded situation. And this situation will be done by monitored by UV absorption here in the UTEC, in the Tecnológica University of Querétaro, and only the shortest double-stranded DNA models will be carried out by NMR in CEITEC. With the new sequences we are proposing, we try to avoid the formation of parallel duplexes. That was an issue we have in the previous studies we have conducted. So this is going to be done, I hope, again by my collaborator, Professor Reinaldo Ples, in the next summer. And we can observe here at the right side typical titration we have done, I think, the last month, and we confirm that the midpoint of this transition is about pH 7.6, which is around the physiological conditions of acidity. So we are going to do some other experiments for the next months. Also, we want to prove this with double-stranded RNA oligonucleotides. Again, by titration monitored by UV absorption and NMR in CEITEC, maybe this year or maybe the next year. And this is the double-stranded model we are proposing with around 17 base pairs, where the uracil groups is using in RNA instead of timing in DNA. So we're interested in conduct these experiments, but now in RNA, according to the relevance we talked some minutes ago in the introduction of this presentation. So another for experiments, we want to determine the protonation enthalpy for, again, a double-stranded DNA model. This time monitored by differential scanning microcalorimetry in CEITEC Brno. Also for the next summer, collaborating with Professor Jeff Hauser and Professor Milka Chitkova in CEITEC, sorry, for the next summer, in order to get the protonation enthalpy of this transition of this protonation of the 5-methylpsilocin. So now we want to extend these studies to biological long DNA model. We have proposed salmon sperm DNA as a first model to be studied for mainly by titration monitored by UV absorption, because they are too long in order to be analyzed by NMR experiments. So it's going to be done, I guess, the next year. Also, as we said before, we want to examine the interaction of model molecule with protonated central base pair, as we see here in this proposed double-stranded DNA model. And we are initially proposing this molecule, host 33258 molecule, which interacts with the major group of double-stranded DNA when the nucleobases are in a neutral state. So if this protonated base pair is present, we are thinking that these interactions will not occur. Okay, this is we're proposing how to prove or test this interaction of double-stranded DNA molecule with a central protonated base pair with a fluorescent molecule that could be represented as a protein that is interacting with the double-stranded DNA. So finally, we want to study by theoretical calculations, again, collaborating with CEITEC, specifically with Professor Peter Kulhanek, and we want we are planning to do by DFT, density functional theory, using the ORCA software and restricted Hartree-Fock algorithm using GAMES software. So similar to these molecular models, we want to study how the energy and structures behaves when this central base pair is protonated or with a positive charge. We have done previously, but with the opposite process, which is the protonation, and we can try some interesting. We have found some interesting energy and structure results. And now we can we are proposing to do it, but now with the protonation, the gaining of a proton in a central nucleotide or a central base pair, depending on the double-stranded or single-stranded model. So these are the future experiments. And finally, I want to conclude with these 3 important points. Due to the biological relevance of the 5-methylcytosine residues in nucleic acids, understanding of these underlying physical processes and related effects are crucial. As we saw in the introduction part, and also the possibility of a positively charged base pair in nucleic acids could explain specific molecular interactions which occur in molecular biology processes. This proposed study will contribute to the biophysical basis of post-translational and transcriptional modifications by methylation of cytosine base groups. So this is the main conclusions for this presentation. These are the references I have used for this talk. And I would like to thank you for your time and your attention. And I'm open to questions. Thank you very much.
Thank you, Julio, for this presentation on the DNA RNA methylations. I don't know if we have questions. I see one for Eugenia, but she told me she cannot see the questions. I can maybe try to answer. And I have one question for you, Julio, to start. You perform your NMR analysis in Brno in Czech Republic. Do you have some collaboration with the NMR Center in Mexico?
No,
with Carlos Amero?
No, because we don't have such advanced instruments as they have in Brno, in CEITEC. They have spectrometers of high capacity. I don't know, the 700 megahertz is a good instrument for these molecular experiments. And here in Mexico, we have, I think we have 2 in Mexico, but they are so expensive to get some experiment there. And for accreditation, we need to do about, I don't know, maybe 10 or 12 different measurements. It need to be too expensive for us.
Okay. And I don't know if we have more questions. I don't see anything. I can try. No. I check in the conversation. I don't know if you can have an access also, Julio, in the Q&A section. If you see something, you try to reply. To be honest,
I'm checking that section.
Okay, I have no more questions.
No, I don't have questions in this section.
Okay, if I can. We have one question for Eugenia, and it was which measurement and experiment did you do in Grenoble? In the second line, we have what are the new findings thanks to this experiment? And Eugenia come back, and maybe she can reply if you want.
Yes, of course. It's the question of Sylvia, right?
Yeah.
Hi, Sylvia. I didn't make any experiment yet. I suppose that I will get the samples soon, so that I can go again to Grenoble or send my samples to make some experiment out there. I hope so. Right, Lionel? I want to see my proteins over there, interaction between protein and metabolite protein, maybe.
Okay, thank you for this answer, Eugenia. I have nothing more from for Julio, I think. Sometimes it's long to come and long to write. We can stay 1 minute more. I want to thank you again for your talks. It was really interesting, and I hope we can organize more and more in collaboration with Brno or in France, in Grenoble, or in other facilities in Europe, and also maybe apply on the facility in Latin America to visit you also. I don't know if Catherine wants to say more stuff about the collaboration, what we can organize. If she's always here, or if John wants to join us to present the next webinar, maybe the 38th. And thank you again, all of you, for the free presentations.
Thank you very much.
Bye-bye.
Have a good day. Bye-bye.
Bye.